All right, so let's talk about free surface modifications, or another way to say this, which I use a lot because this is the way I was trained, apical surface modifications. Right, so these are things that are being changed to basically your top cells to undergo different functions. Okay, so if the top apical side of the epithelial tissue is smooth, it's mainly there for reducing friction. So like endothelium tissue types, right? Endothelial layers and blood vessels. If there are folds in them, like in your transitional epithelium, you need to change shape, like the urinary bladder, so it can stretch, basically, is what's indicating there. You can have microvilli, which increases the surface area for absorption or secretion. Now, you could also have specialized microvilli that are now becoming more sensory receptors, like your stereocilia in your inner ear. They're not cilia, okay? They're slightly different, and they actually transfer physical sound vibrations into electrical signals, okay? So that's what those microvilli are doing. Then you have your cilia, which are moving mucus or other things across the surface of the cell, right? It's helping move things across the cell, but it's not moving the cell. So these are modifications that you can do to your top apical or free surface of your epithelial tissue. Now, cell connections, remember, epithelial cells love having neighbors. They are the extroverts of the tissue types, okay? So they love their neighbors, and because of that, they have different types of connections, and therefore have different functions. They have these functions from the connections. They are found on the lateral side and the basal surfaces of the cell. They are there to form a permeable layer, as in your tight junctions here. They are there to bind cells together via adhesion and desmosomes. They provide mechanisms for intracellular communications between cells, right? Because they're like little portholes to send signals through, right? As well as hemidesmosomes to bind or integrins that bind the cell to the basement membrane. Okay, so you can have desmosomes or hemidesmosomes, like semicircle, tight junctions, adhesions, and gap junctions, which is shown here, right? And that's where your signals are being transferred through. So inside your cell connections, desmosomes, they're disc-shaped regions in the lateral side of the cell. They're really there when you're having the cells exposed to a lot of stress. So think of your epithelial cells lining your mouth, vaginal cavities, anything undergoing lots of friction. These are there to basically hold the cells together. You have a hemidesmosome cell connections. These are connecting the epithelial cells to the basement membranes, right, to the extracellular matrix to prevent the movement of the tissue. You don't want the sliding unless you want it to. Now, the tight junctions are there to hold the cells together, and this is really forming that permeable membrane in between the cells. We're not talking about the cells being permeable themselves. We're talking about that gap between cell to cell. You want to prevent permeability as much as possible because you want it to be regulated going through the cell and not unregulated going through the spaces in between the cells. Then you have adhesion connections. These are right below the tight junctions, and these are really connecting epithelial cells to one another, and also preventing passage of materials, larger materials between, right? And then further strengthening of that connection. They're just different proteins. That's why we're classifying them as different, and they have different functions outside of just cell connections here, more related to like cellular communication, which we're not going to get into because you guys, that's a cellular biology class concept, and you don't need to worry about that. Gap junctions are channels, right, integral membrane proteins that bind to other gap junctions and other cells to allow ions and small molecules to pass through. We don't know exactly their full function in the epithelial cell tissue type, but we do know what they do. We just don't know why they do it. Now, in cardiac cells and like muscle cells, they actually allow electrical signals to pass from cell to cell, which is good because you want the contraction signal in the heart to move, from the top of the heart to the bottom of the heart, right? So that's important there, and that's an example of why you would have that. And then they may help move cilia, again, not exactly sure. Here's a funny cartoon kind of, highlighting the different types, and maybe that helps. Just so you're aware, plasma desmata is not in animal tissue, right? That's only in plants, okay? So that is not an answer on an exam or quiz or homework. No. That is a cell junction, but that is not in animals, so do not select that, okay? Now, glands are a fun little example of epithelial cells. You wouldn't necessarily, tissue types, I should say. They're not exactly what you would think that they would be epithelial, but they are in support of the connective tissue, and that's what that CT is there for. There are two different types of glands because they're infolding of the epithelial tissue, and they're surrounded by and organized by what they look like, their structure. So endocrine glands, they're not in open contact with their exterior, right? There's no ducts, there's no blood, no ducts connecting them, touching all the cells, but they do have blood vessels kind of surrounding them, is the idea here, where you can produce hormones, and that's the main idea here. So I'll go to exocrine here. So exocrine, they have like a duct or open void connecting all of the apical side or the free surface side of the epithelial cells. Like exocrine examples of this is like mucus secretion or milk secretion in mammary tissue, right? Those are exocrine glands. Endocrine glands, like the endocrine system, hormone systems, those are enclosed. So there's no duct going into the center of the gland, but there are blood vessels surrounding them that then the hormones are secreted into the blood and then shipped out through the rest of the body. So there are ways to further classify glands. There are simple glands, which are just, tubular in form, no branches. There are, they're still kind of multicellular, but there are simple branched tubular glands, right? So they're multiple branches but are tubed. There are acinar glands, like the sebaceous glands of the skin or milk glands in the mammary tissue. They have more sac-like. And then there's, of course, simple branched acinar, or acinized, the plural form of that glands there. So kind of straightforward there, not too bad, but they're all multicellular, so they have multi-glands there. Now you can have compound glands, so they're multicellular again, but they have many branches, right? So maybe you have part of it tubular, part of it acini, or both, right? And examples here, again, mammary glands are a great example of this, or your pancreatic glands for digestion, right? So compound tubular, compound acinar, or compound tubulo-acinar, just examples of those, right? So you can have large branching of these. Now, exocrine glands, you could have merocrine glands where they exocytose, which is the most common. So they, use exocytosis to ship the hormones out. You could have apocrine glands where they actually pinch off fragment of the cells, right? So this is slightly different than exocytosis, remember, because exocytosis, you packaging in membrane-bound vesicles and shipping out, versus apocrine glands, you're actually pinching off fragments of the cells themselves and sending that out. So like mammary glands and seromyngous glands. Now, holocrine exocrine signaling is where the entire cells are pulled off, right? So sebaceous glands here, right? Totally getting sent out here in your exocrine gland signaling, right? So there's, again, further classification of your exocrine signaling going on here. And this is in the ducts, right? So we talked about the organization of the ducts for exocrine signaling, and now we're talking about functionally how do they secrete their molecules and compounds, either freely via exocytosis, part of the cell, apocrine glands, or the whole cell. And here's kind of image here of like our simple and stratified tissues. This is what we would call histology, right? Imaging of the different tissue types themselves. Moving on to connective tissue here. So this is another tissue type. It's very abundant. It's found in every organ, right? Because everyone needs to be attached, right? They are consistent of cells separated by extracellular matrix, right? So that's a difference from epithelial where they're touching one another. Connective tissue are separated from another by extracellular matrix. So they are very diverse in their functions and the types. So functions here, they enclose organs, so your fasciae, and they separate organs into different layers. They can connect other tissues to one another, tendons and ligaments. Movement, your bones, storage as in fat. Another, insulation and cushioning is fat, for example, again. Transport your blood. Look, it's a connective tissue. It connects your body to oxygen. You can think about it that way. And protect immune cells. So there are different words that we use to classify the cell types. So descriptive stem words, blast, so osteoblast, like down the bottom there, they create the matrix. If you see osteocytes or chondrocytes, they are maintaining the extracellular matrix. Or class, they are breaking down the matrix. So fibroblasts, osteoblasts, they are exploding. I don't know, that's how I kind of remember it, is class explosions. I don't know why that makes sense to me, but it does. But those are, when you see those things at the end of a tissue cell type or tissue type, that kind of tells you what their function is inside of the connective tissue. So like osteoblasts, they form bone, which then they can also become osteocytes, which then maintain the bone. An osteoclasts or blasts, class cut away, they break bone down. Chondroblasts form cartilage, which then chondrocytes maintain it. Fibroblasts, on the other hand, produce fibrous tissue, which then fibrocytes maintain that tissue type. So here are other types of cells in our connective tissue family. Adipose cells, which are our fat cells, really dermis cells, skins, and it's also in your bone marrow. That's another place you'd find it. Mast cells, which are kind of along your small blood vessels related to inflammation. Your white blood cells, your macrophages, which are phagocyzing other objects and cells. Platelets, our stem cells, which are called mesenchymal stem cells when we're talking about connective tissue. They differentiate into all these things like this figure shows here, right? For my doctorate, I highly studied mesenchymal stem cells. So that's my go-to cell type. We will pause the video there. We'll move on to the next one because we'll move into what is the extracellular matrix and what is it made of. So I will see you in the next video.